Welcome to the PubMed Daily Review for March 11th. Today we scanned 6,343 new open access papers. Here are the top 5 selected for detailed analysis. Up next, our first paper out of 5, Plasma Proteomic Dynamics Preceding Glaucoma reveal a 15-year pre-diagnostic window. Causal insights and predictive utility in 45,850 participants by Inka Zhao and Jihong Wu from Department of Ophthalmology, Eye&ENT Hospital, College of Medicine, Fudan University, Shanghai, China. Let's break down its key contributions. Glaucoma is a silent thief of sight, often progressing unnoticed until irreversible damage is done. The challenge lies in detecting this complex disease in its earliest stages, before vision is lost. This study aimed to uncover if subtle changes in our blood proteins could act as an early warning system, potentially giving US years of advance notice. Researchers analyzed blood samples from over 45,000 individuals in the UK Biobank, none of whom had glaucoma at the start. They measured the levels of nearly 3,000 proteins in their plasma over a median follow-up of more than 16 years. The goal was to see which proteins were linked to developing glaucoma and if these changes could predict the disease years in advance. The study found 136 proteins significantly linked to glaucoma risk, with EDA2 showing a notable association. Excitingly, these proteomic changes began appearing 12 to 15 years before a clinical diagnosis. Furthermore, machine learning models using these protein signatures achieved an impressive AUC of 0.803, significantly outperforming models based on demographics alone. This research opens a remarkable 15-year window for potentially detecting glaucoma before symptoms even appear, using simple blood tests. The discovery of causal proteins like LTBP2 not only deepens our understanding of how glaucoma develops but also points towards new avenues for treatment. Ultimately, these findings pave the way for innovative screening strategies to identify at-risk individuals much earlier. Up next, our second paper out of 5, Artificial Intelligence, Enabled Multi-Omics Biomarkers for Immune Checkpoint Blockade, Mechanisms, Predictive Modeling, and Clinical Translation by Xiaodong Wang and Qianqian Wang from Department of Oncology, Zhuzhou Hospital affiliated to Xiangya School of Medicine, Central South University, Zhuzhou, China. Let's break down its key contributions. Immune checkpoint inhibitors, or ICIs, have been a game-changer in cancer therapy, leading to remarkable remissions in some patients. However, a significant challenge remains: many patients don't respond, or their response is short-lived. This is partly because single biomarkers, like PD-L1 expression, just don't capture the full picture of the complex tumor immune system. That's where multi-omics profiling comes in, offering a much deeper dive into the biological factors at play. This review explores how artificial intelligence, or AI, can be used to make sense of vast amounts of data from different biological sources, what we call multi-omics. By combining information from genomics, transcriptomics, proteomics, and even imaging and microbiome data, AI can build sophisticated models. These models aim to predict which patients are most likely to benefit from ICIs, moving beyond the limitations of older, single biomarkers. The results are promising. For instance, in lymphoma, an AI model integrating multiple data types achieved an impressive area under the curve of 0.94 for predicting response to anti-PD-1 therapy, far exceeding the performance of individual markers. These AI-powered composite biomarkers are proving much more effective at capturing the complex biology that dictates whether a patient will respond to these powerful treatments. So, what does this mean for the clinic? AI-enabled multi-omics represents a significant leap forward in personalizing cancer immunotherapy. While challenges like data standardization and rigorous validation need to be overcome, the future looks bright. Continued research into advanced AI techniques and data integration promises to unlock even more precise and effective treatment strategies for our patients. Up next, our third paper out of 5, Application and Progress of Artificial Intelligence Agents in Drug Development, by Donghai Zhao and Changyu Si. Let's break down its key contributions. Drug discovery is a long, expensive, and risky process. We need new ways to make it faster and more accurate. Artificial intelligence, especially AI agents, are emerging as a powerful new approach. These agents can reason, plan, and use tools autonomously, acting as collaborators rather than just tools. This review explores how AI agents are being applied across the drug development pipeline. These agents use advanced AI to reason and plan, integrating various research stages. They can help identify new drug targets, design novel molecules, and even automate the process of synthesis and experimentation. The progress of AI agents in drug development is impressive. They are successfully identifying new targets and mechanisms by integrating vast amounts of knowledge. Furthermore, they are automating complex tasks like molecular design and synthesis planning. This allows for a seamless connection between virtual design and physical experiments, paving the way for more advanced applications. In conclusion, AI agents represent a significant leap forward in drug development. They are becoming indispensable collaborators, accelerating the discovery process. While challenges remain, the future holds promise for even more integrated and powerful AI systems in medicine. Up next, our fourth paper out of 5, CRISPR-based diagnostics for infectious diseases, mechanisms, advancements, and clinical transformation prospects by Xinjun Pan and Fengren. Let's break down its key contributions. Infectious diseases continue to challenge global public health, demanding faster and more accurate diagnostic tools. Traditional methods, while useful, often fall short in critical situations like emerging outbreaks or when rapid point-of-care testing is needed. This is where the revolutionary CRISPR-Cas technology steps in, offering a new paradigm for infectious disease diagnostics. CRISPR-based diagnostics harness the precision of CRISPR-Cas systems, primarily Cas12 and Cas13, for detecting infectious agents. These systems work by first recognizing specific pathogen nucleic acid sequences. Once bound, they activate a unique collateral cleavage activity, which then amplifies a signal by cleaving many reporter molecules. This dual action allows for incredibly sensitive and specific detection. Key advancements have led to remarkable performance metrics. These systems can achieve attomolar sensitivity, meaning they can detect incredibly small amounts of pathogen genetic material. Furthermore, detection can be achieved in as little as 5 to 60 minutes, a significant improvement over traditional methods. Prominent platforms like Sherlock and Detector, utilizing Cas13 and Cas12a respectively, employ various readout methods including fluorescence and lateral flow assays, making them adaptable for different settings. The clinical transformation prospects for CRISPR diagnostics are substantial. They offer a powerful new tool that could revolutionize infectious disease management by providing rapid, sensitive, and portable testing. However, challenges such as regulatory hurdles, cost, and differentiating live from dead pathogens need to be addressed. Continued research and development are crucial to fully realize the potential of these technologies for global health. Up next, our fifth paper out of 5, The Augmented Physician, AI and the Future of Clinical Cognition by Caradine Buabada and Enoch Anane from Yukon Health, Farmington, Connecticut, United States. Let's break down its key contributions. Medicine is facing a cognitive crisis. The amount of new biomedical information is exploding, doubling every few months. This makes it impossible for any single physician to keep up, threatening the very foundations of clinical expertise and patient safety. We're seeing a widening gap between what we know and what we can actually apply in practice. This paper takes a prospective approach, examining how artificial intelligence can help us navigate this information overload. It argues that AI shouldn't replace doctors, but act as a cognitive partner. Think of it as a tool to help organize and contextualize the vast amount of medical data, allowing physicians to focus more on critical thinking, empathy, and patient connection. The evidence is mounting. Studies show AI-assisted documentation tools can significantly reduce workload, with one meta-analysis showing a standardized mean difference of -0.72. In a real-world study, an ambient AI scribe reduced burnout from over 51% to under 39% and saved clinicians nearly an hour of documentation time daily. Overall administrative burden reductions are exceeding 30%, showing AI's tangible benefits. So, what's the takeaway? The future of medicine isn't about AI replacing doctors, it's about augmenting them. AI offers a vital solution to the overwhelming information challenge, helping to reduce burnout and improve efficiency. The key is a thoughtful human-AI partnership, ensuring technology supports, rather than supplants, the core human elements of care.